Hey guys, hi, welcome to once again a PYQ video where we are going to discuss question number 9 of the 2013 IBO paper. Okay, this question is about the regulation of the cell cycle. Okay, now whenever you get something this complicated, please always try to read the question and gather information from there rather than trying to remember the whole concept in your head, because most of the times the question provides you with all the information. You have to deduce, you have to make an informed choice. Okay, so progression through the cell cycle is mediated by cyclin dependent kinases. So kinases, you understand, that means it's going to do something about the phosphate group. It's going to add or remove a phosphate group. So these are proteins and they are dependent. Okay, so these kinases are dependent on another protein, which is a cyclin. Now why the name is cyclin? Because the concentration of this protein changes. Okay, so in a cyclic fashion, that means sometimes it goes up, sometimes it goes down, which become active when bound to their respective cycling and okay, so CDKs are active when they are bound to their respective cycling. So that is why they are say dependent, right? Quite clear. And phosphorylated at the threonine C or core threonine. Now remember, the CDKs, it's a protein, that means it's a chain of amino acids and it will have plenty of amino acids. Certain times when you phosphorylate a particular amino acid, it may activate or deactivate the particular protein. In this case, they mention that phosphorylation at threonine C activates it. Okay, so active when phosphorylated at threonine C. Okay, good. Phosphorylation or dephosphorylation of other amino acids further modulates. So by modulation, it means that they are not explicitly saying whether it increases, decreases, activates, inactivates the activity. They are not saying that. It just modulates and modifies, changes somehow. The following pathway represents the protein involved in the entry into the M phase of the cell cycle. Now, this is where the trick comes in, because somehow you're not very familiar with what's happening. So let's first read the chart. So phosphorylation, addition of a phosphate, may either activate or inhibit, right? So there is a legend. If it's like this, that means this phosphorylation is activating something, or if it's going to inhibit, that's going to be like that. Now, at the same time, dephosphorylation, removal of a phosphate group, it may also activate something or deactivate something, right? So we have to look at what's happening here and then answer the questions. Okay, what you can also do is try to read the prompts and then analyze it, or the other way around, whichever suits you. I usually prefer to read the prompts because I want to work efficiently with time. Okay, but just for the sake of understanding, let's also read this. So there are two different cyclins. We can clearly see there's cyclin H and cyclin B. Okay, not a problem. There are two different CDKs, cyclin dependent kinases, which will be active when the respective cyclin binds. Okay, CDK7, CDK1. Bingo. Now, the CDK7 has a threonine C. Okay, and the CDK7 sort of phosphorylates itself in a way at threonine C, or it may phosphorylate the threonine C of the CDK1. And because of the nature of the arrows shown here and the legend, in both the cases, it's activating it, right? So let me color them green. So this is phosphorylating it and activating it. Okay. Now, something called PP2A, when it does something to threonine C, we have to understand first what is this. So this is inhibiting. So let's draw a red line. Okay. And this is dephosphorylating it, and this dephosphorylation inhibits it. Okay, cool. Sort of try to not create your own legends here. I did that, but okay, we shouldn't try to add more information. It will just jeopardize your own confusion between the questions. Okay, similarly, what is happening here with CDK1 and V1? So CDK1 does something to the V1. As you can see, it phosphorylates it, and this phosphorylation is inhibitory. Okay, and the V1 once again phosphorylates tyrosine 15, and this phosphorylation is also inhibitory. MIT1 can phosphorylate TIR15 tyrosine at 15th position, or threonine at 14th position, and in both the cases it's inhibiting. Okay, now we have CDC25. CDC25 can dephosphorylate tyrosine 15 or threonine 14, and in both the cases it's activating it. Okay, so with all of this done, if we have the activated CDK1, if the CDK1 is active, the M phase, that's bingo, that's where it goes. Okay, now if you're slightly confused, I would want to remind you of the phases of the cell cycle. That's G1, where the cell grows, S, where the duplication of DNA happens, G2, and the M phase. Okay, the checkpoints where the cell checks is it okay to move ahead are found in G1. They're found in the G2M transition, and finally in the metaphase to anaphase transition. These are various points. They have various mechanisms. This is just one of the mechanisms the question is talking about. Okay, cool. Now let's read the questions and answer them one by one. Indicate for each of the following mutations if it would promote entry into the M phase. Okay, so we have to understand if I have an active CDK1, because as per this, an active CDK1 means entry into the M phase. Okay, that's our question. If the activation of CDK1 complex is there, I will be entering the M phase. A mutation that reduces the dephosphorylating activity of CDC25. So that's our guy, CDC25. What does it do? It, according to the legend, dephosphorylates threonine 14 and tyrosine 15. But both of these effects are activating in nature. Okay, the dephosphorylation activates it, but the mutation is reducing the effect. So it's reducing the activation that's happening. That means this is not promoting the entry. So this is going to be a false statement. Okay, pretty easy, right? A mutation that reduces the phosphorylating activity of V1. So here's your guy V1. What does V1 do? It inhibits by phosphorylating tyrosine 15. So it was an inhibitor, right? And it was an inhibitor by phosphorylation. And that is what we are doing. We are reducing the phosphorylating activity of V1. Okay. So if you reduce the phosphorylating activity, which was inhibitory in nature, you're reducing the inhibition. That means CDK gets to be active. That's good for us. That's what the question wants. So this is true. This will promote the entry. Next, we have the third statement. A mutation that changes the threonine C of the CDK1, threonine C of the CDK1, okay, to a valine that cannot be phosphorylated. Now, this threonine C is being activated by CDK7, and it's also, let me erase this, to just remove our confusion. Okay, no, not a problem. So, if you are changing this threonine C, the activation from CDK7 will not be there, which is something that we don't want, right? Hence, once again, I can claim that this is a false statement. Okay, come to the next. A mutation that inhibits the binding of cyclin H to CDK7. See, once again, the CDK7 activates the CDK1 by phosphorylating the threonine C, right? So if we are not able to, if the CDKH and cyclin H, cyclin H, CDK7 do not bind, then the promotional activity will not happen. Okay, that means no active CDK1. So once again, this will also be a false statement, right? So pretty easy, right? I hope this helped. Guys, do remember to share this with your friends. Also, do check out the NCERT map. It's an amazing book. Lots of images, lots of illustrations, lots of amazing stories to help you connect the concepts like a pro. Okay, get your copy from Amazon, or you can access more of these things from my website shantasonasingh.com, or simply check the YouTube channel. You'll find plenty of resources to help you prepare for your Olympiads. Take care. See you in the next one.